Imagine a time when our planet was utterly alien. Picture a molten, storm-swept world, completely devoid of life. I mean, no rustling leaves, no chirping birds, not even the simplest microbe.
Nothing we'd recognize as living, just rock and gas and heat.
Yet somehow, from this incredible chaos, something truly extraordinary emerged. Life itself. This isn't just a captivating story,
Yeah.
It's one of science's most profound and enduring mysteries.
Oh, absolutely. It's a question that has fascinated thinkers for millennia. But it's really only in the last century or so that we've begun to tackle it with the kind of scientific rigor needed to move beyond just, well, speculation.
And that's exactly where we're headed today. We're taking a deep dive into abiogenesis, the scientific journey to understand how life spontaneously arose from non-living matter right here on Earth.
Yeah, the nuts and bolts of it.
We'll explore the incredible evidence and the cutting-edge theories that scientists are using to piece together this epic, almost cosmic tale.
And it's vital, right from the start, to make a key distinction. It's one that often causes confusion, actually. This isn't the old, disproven idea of spontaneous generation.
Right, like maggots from meat.
Exactly. That old idea incorrectly claimed that complex creatures could just pop into existence from decaying matter, whenever. That's a myth, completely debunked by scientists like Louis Pasteur centuries ago.
Okay, so biogenesis is different. How?
Abiogenesis, in contrast, proposes a gradual step-by-step process. It likely happened just once or maybe a few times under the utterly unique, really extreme conditions of the primitive Earth. Conditions that, remarkably, simply don't exist today.
That's a crucial clarification. So our mission today, then, is to understand how Earth's raw energy and chemistry conspired, essentially, to create the fundamental molecules of life.
The building blocks.
Yeah, lipids for membranes, carbohydrates for structure and energy, amino acids for proteins, and nucleic acids like DNA and RNA for genetic information. And then how these molecules somehow organize themselves into the very first cells,
The first containers for life's processes.
eventually leading to all the incredible diverse life we see around us. You're going to discover how geology, chemistry, and biology all weave together to bring a plausible, though I guess still incomplete, narrative into focus.
It's definitely still incomplete, yeah. We'll begin by setting the primordial stage, exploring the truly hellish conditions of the early Earth. Then we'll examine groundbreaking experiments that help kick off this entire field of research, like the famous Miller-Urey experiment.
Ah, yes, Miller-Urey. We'll definitely get into that.
And then we'll look at the competing cradles where life might have truly begun.
After that, we'll grapple with the mind-bending emergence of the very first cells, how they evolved, and finally we'll look at the physical fingerprints life left behind in ancient rocks, including, arguably, the most significant planetary transformation ever engineered by biology.
It's a huge story.
It really is. It's a journey from stardust to stromatolites, a story that truly unpacks Earth's first spark. Let's dive in. Okay, so to truly appreciate this spark of life, we have to travel back, way, way back, to what scientists call the Hadean Eon.
The very beginning.
This was the earliest period of Earth's history, spanning from about 4.54 down to 4.0 billion years ago. And it's named after Hades, the Greek underworld, for a very good reason.
For a very good reason.
Precisely.
When Earth first formed, roughly 4.54 billion years ago from the accretion of cosmic dust and gas, it was almost unimaginably violent. The sheer energy released from gravitational accretion, combined with constant cataclysmic collisions with other space debris, kept the entire surface of the planet in a completely molten state.
Molten? You mean like liquid rock everywhere?
Picture a global ocean of magma, yeah. Constantly churning, glowing red hot. Volcanic activity was unimaginable compared to today, constantly spewing gases.
Wow. A global ocean of magma. That's hard to even picture. Given that immense energy, what kind of geological processes were shaping the planet back then, besides just cooling down?
Well, a major event happened around 4.5 billion years ago. There's compelling evidence of a colossal collision with a Mars-sized protoplanet, often called Theia.
Theia, right, the moon-forming impact.
Exactly. This impact was so powerful it's believed to have blasted enough material into orbit to form our moon, and in doing so, it completely remelted Earth's surface again, basically hitting the cosmic reset button.
So any progress towards cooling was wiped out.
Pretty much. And any initial thin atmosphere of maybe hydrogen and helium that Earth might have captured from the solar nebula, that would have been stripped away entirely by this cataclysmic event.
So a truly hellish landscape, constantly being remade by these giant impacts. But even this eventually had to cool down, didn't it? What happened as the planet began to stabilize, even a little?
It did, but incredibly slowly. Over the next few 100 million years, the planet began its very slow process of cooling. This allowed for the gradual formation of a thin, solid crust. But it was constantly being recycled, broken up, melted again.
So finding rocks from that time is almost impossible.
Extremely difficult. The continuous recycling of the crust wiped most of it away. The only direct physical evidence we have are these incredibly resilient little crystals called zircons. Some of them date back as far as 4.4 billion years.
Zircons, okay. Tiny time capsules.
Kind of, yeah. And what's truly surprising is that even these ancient zircons hint, surprisingly, at the presence of both a solid crust and even liquid water very, very early on.
Liquid water that early. That seems almost counterintuitive for such a fiery beginning.
It does, but the zircon chemistry suggests it. And that sets the stage for the next crucial element, the atmosphere. Because the sky above this nascent Earth was definitely not blue. It was, well, alien.
Right, so what was it like?
As Earth continued to cool and differentiate, heavier elements sinking to the core, lighter ones rising, its first stable atmosphere began to emerge, mainly from continuous volcanic outgassing. Just volcanoes pumping gas into the sky constantly.
And vastly different from today, I assume.
Oh, completely. A geochemical model suggests this atmosphere was likely neutral or maybe weakly reducing.
Okay, you mentioned a neutral or weakly reducing atmosphere.
Yeah.
For us, breathing oxygen, that sounds incredibly alien. What did that specific atmosphere composition mean for the chemistry happening on this early Earth, especially for the raw materials for life?
It meant it was rich in molecules that readily donate electrons, think things like hydrogen, maybe some methane, ammonia, which is crucial for building complex organic structures from simpler inorganic ones. And critically, it contained almost no free molecular oxygen, 0 O2.
No oxygen at all. What were the main gases then?
The dominant gases were immense quantities of water vapor, nitrogen, which is still the main component today, actually, and carbon dioxide, huge amounts of CO2.
How much CO2 are we talking?
Estimates suggest CO2 levels were maybe 10 to even 200 times higher than they are now.
Whoa. That's an incredible amount. That must have created quite a greenhouse effect, right? Keeping the planet from freezing solid, especially with a younger, fainter sun.
It absolutely did. This dense CO2 blanket was essential. It kept the planet warm despite a significantly fainter young sun, maybe 30% less luminous than today. There were also smaller but chemically important amounts of hydrogen, carbon monoxide, methane, ammonia, and hydrogen sulfide present.
But the key takeaway is almost no free oxygen.
Virtually none. And this anoxic condition was absolutely essential for abiogenesis because those simple organic molecules that are life's building blocks, they're incredibly fragile. They would have been rapidly oxidized, basically burned up or broken down by oxygen. Life simply had to begin in an oxygen-free world.
So, atmosphere without oxygen. But that also meant no ozone layer, right? Which protects us from UV radiation today.
Exactly. No O2 means no O3, no ozone.
And that brings us to what's often called the UV paradox. The very energy that might have helped spark life was also a potential death sentence for it.
Yeah.
That's a profound dilemma.
It is.
What does that tell us about where life couldn't possibly have begun and where it must have found refuge?
That's the core of the paradox, yeah. With no protective ozone layer, the surface was just blasted by intense, high-energy ultraviolet radiation from the sun. Now, UV radiation, along with lightning and geothermal heat, was a necessary energy source to drive the synthesis of the first molecules. You need that energy input.
Right.
But it was also incredibly destructive to those very same molecules. It could break them apart just as quickly as they formed. This strongly implies that the crucial early chemistry needed to happen in sheltered microenvironments.
Like where? Underground? Underwater?
Maybe deep within the water column, or inside the tiny pores and crevices of rocks, or perhaps near deep-sea hydrothermal vents, places where molecules could be synthesized using available energy, and then immediately protected from the very energy source that created them. It suggests a very specific set of, well, Goldilocks conditions were needed, not just anywhere.
Speaking of water, you mentioned the first oceans earlier. When did those finally form and what were they like? Because I imagine they weren't exactly pristine blue seas.
Far, far from it. As the surface finally cooled below 100 degrees C, the boiling point of water, the vast amount of water vapor that had built up in the atmosphere, condensed. This triggered a great deluge, raining down for potentially millennia.
Wow, rain for thousands of years.
Yeah, eventually forming Earth's first oceans around maybe 4.3 billion years ago. This was a pivotal moment. It provided the liquid medium essential for life's chemistry, and it also helped regulate the climate by dissolving some of that abundant atmospheric CO2.
So the oceans help pull some of that CO2 out of the atmosphere, acting as the planet's first sort of carbon sink.
Exactly. But these primordial oceans were still a chemically potent and pretty harsh brew. Because of the high atmospheric CO2, they were likely more acidic than today's oceans, with a pH possibly between 6.0 and 7.5 compared to around 8.1 now.
So acidic. Hot. What else?
They were also hot, probably much warmer than today, and salty, and absolutely full of dissolved minerals leached from all that volcanic crust. Intriguingly, they likely had high concentrations of dissolved ferrous iron Fe2 plus Sela, which is soluble in anoxic water but precipitates out in the presence of oxygen. And they were probably poor in sodium but rich in potassium.
That's an intriguing point about the chemical signature low sodium, high potassium. Is it fair to say then that the internal chemistry of our own cells still carries a kind of molecular fossil of those ancient conditions? Because that matches cell chemistry pretty well, doesn't it?
It's a very striking parallel, yes. Many scientists think so. The internal environment of virtually all modern cells mirrors that low sodium, high potassium ratio. It's one of those beautiful convergences in science that hints at the deep history we're discussing.
Amazing. Okay, so we have these hot, acidic, iron-rich oceans. But you also mentioned that just having a water world wasn't enough. The emergence of the first continents, even just as scattered volcanic islands, is now seen as critical. Why?
While the deep ocean offered protection, these land-water interfaces, think shallow pools, new volcanoes, wetlands, geothermal hot springs on land, offered two huge advantages over the vast, deep ocean.
Okay, what were they?
First, concentration. Evaporation in shallow pools could concentrate those chemical precursors, making the soup much thicker and more reactive. It allows molecules to actually find each other and interact much more easily than when they're diluted in a massive ocean.
Makes sense. More bumping into each other.
Right. Second, and perhaps even more powerfully, these environments created wet-dry cycles.
Wet-dry cycles. You mentioned these being crucial for building bigger molecules. Can you elaborate on how that works chemically?
Absolutely key. These cycles are powerful drivers for polymerization. That's the linking of simple monomers, like amino acids or nucleotides, into the long, complex chains life needs, like proteins and RNA. The chemical reaction that forms these links is called dehydration synthesis because it releases a water molecule.
Okay. Dehydration, removing water.
Right. Now, trying to remove water in water, like in the middle of the ocean, is thermodynamically unfavorable. The overwhelming tendency is for the reverse reaction, hydrolysis, the breakdown of polymers, by adding water to occur. Water wants to break the chains, not form them.
So the ocean is actually hostile to building polymers.
In a way, yes. But on the edge of a pool, as it dries out, water evaporates, and that physically removes one of the products of the dehydration reaction. This pulls the reaction forward, strongly favoring the formation of long chains. Then, when it rains or the pool refills, these new polymers can move around, interact, maybe fold up. This suggests that the most productive zones for creating life's complex polymers weren't the deep ocean, but these dynamic, sometimes messy interfaces between land and water.
Okay, so we've got this picture now. A hellish Hadean Earth, slowly cooling, getting its first oceans in this strange oxygen-free atmosphere, and crucially, these dynamic land-water interfaces where chemistry could really start getting interesting.
The stage is set.
Exactly. With that stage set and the first ingredients swirling around, the scientific journey to figure out how life started truly began to heat up. And that brings us directly to perhaps the most iconic idea in origins of life research, the primordial soup.
The primordial soup, it's such a great mental image, isn't it? And it really captures the essence of the Oparin-Haldane hypothesis, which was truly revolutionary for its time back in the 1920s.
Oparin and Haldane, right. Two scientists working independently.
Yes, Russian biochemist Alexander Oparin and British scientist J.B.S. Haldane. They independently proposed that early Earth conditions, specifically a reducing atmosphere, meaning one rich in electron-donating molecules like methane and ammonia, and crucially lacking free oxygen, were the key.
So they imagined this chemical stew basically being zapped by energy from the environment.
Precisely. They theorized that energized by things like lightning and the intense UV radiation we talked about, simple inorganic molecules in the atmosphere and oceans could react to form various simple organic molecules, what we call monomers, amino acids, simple sugars, things like that.
And Haldane specifically coined the soup term.
Haldane famously described these compounds accumulating in the early oceans over millions of years, creating this a hot, dilute soup or prebiotic broth. Within this soup, these monomers would then bump into each other, interact, maybe link up, polymerize, and gradually assemble into more complex structures, eventually giving rise to the first self-replicating, metabolizing entities. Life, basically.
That was huge. It provided a testable scientific pathway from non-life to life, didn't it? Moving it out of philosophy and into the realm of chemistry.
Absolutely. It framed the question in a way scientists could actually investigate.
And the experiment that truly put this theory on the map, that made it tangible for everyone, was the Miller-Urey experiment in 1952. It's hard to overstate what a landmark study that was.
Oh, it was monumental. It utterly transformed the field. For decades, Auburn Haldane was just this compelling idea. But Stanley Miller, who was then a graduate student working under Nobel laureate Harold Urey at the University of Chicago, designed this ingenious experiment to actually simulate those proposed conditions in a closed glass system.
Okay, for those who might not recall the specifics, maybe describe the setup for us. What did it look like? It's quite iconic.
It really is. Picture a compact, almost elegant setup of interconnected glass flasks and tubes. He had a boiling flask at the bottom that represented the ocean, providing water vapor.
Okay.
This vapor then circulated up into a larger flask that served as the atmosphere. Inside this flask, he put a mixture of gases thought to represent the early reducing atmosphere methane, CH4, ammonia, NH3, and hydrogen, H2.
The gases Oparin and Haldane proposed.
Exactly. Then, crucially, Miller and Urey discharged continuous electrical sparks between two tungsten electrodes inside that gas chamber, vividly mimicking primordial lightning storms.
Zapping the atmosphere.
Zapping the atmosphere. Then a condenser cooled the mixture, allowing any newly formed compounds to condense like rain and collect in a U-shaped trap at the bottom, simulating accumulation in the ocean, preventing them from being destroyed by the sparks immediately after forming.
So ocean, atmosphere, lightning, rain, all in a closed loop. And what were the results? What did they find after just a week of running this?
The results were, well, truly stunning. After just 1 week, the water in the trap had turned a deep red color and was coated with this brownish-black tarry substance. Miller's careful analysis revealed that a significant portion of the initial carbon from the methane had been converted into new organic compounds.
And the big discovery.
Most importantly, he identified several amino acids, including glycine, I-alanine, and valine. These are the fundamental building blocks of proteins, the workhorses of all life.
To synthesize life's building blocks in a flask like that, it must have been monumental. What was the immediate scientific reaction to Miller-Urey, and how did it change the direction of origins of life research almost overnight?
The impact was immediate and profound. This experiment provided the first concrete experimental evidence that life's essential molecules could indeed arise from simple non-living chemicals under plausible natural processes. It dramatically demonstrated that the perceived gap between inorganic chemistry and organic chemistry, the chemistry of life, was not impassable, as many had thought.
It made abiogenesis seem possible, even likely.
Exactly. It launched the modern experimental field of origins of life research, moving it from pure theory into the laboratory. And it lent powerful initial support to that primordial soup hypothesis. Research funding flowed and new labs started up. It really ignited the field.
So Miller-Urey seemed to prove the soup hypothesis pretty convincingly at the time. But science, as always, evolves. What criticisms or challenges did the experiment face over the following decades?
Right, no experiment is perfect, especially one trying to simulate something billions of years ago. Despite its huge success, the Miller-Urey experiment faced scrutiny, primarily concerning the exact composition of the simulated atmosphere he used.
The methane and ammonia.
Yes. As our understanding of planetary formation and early Earth geology improved, modern geochemical evidence began to suggest the early atmosphere was more likely neutral, dominated by carbon dioxide, CO2, and nitrogen, N2, maybe with some hydrogen, but not the strongly reducing methane-ammonia mixture that Miller used. That kind of atmosphere is harder to maintain geologically for long periods.
Okay, so a less reactive atmosphere.
I know.
And when later experiments repeated the setup but used that more realistic CO2-N2 mix, what happened to the yield of organic molecules? Did they still get amino acids?
Well, initially, experiments using that more realistic CO2-N2 mixture yielded dramatically fewer organic molecules, especially amino acids. The yields were much, much lower, and that was seen as a significant challenge to the original interpretation and the whole soup model.
So did that kill the idea?
Not quite. The story, as often happens in science, became more complex and actually more interesting. Later research showed that adding common early Earth minerals into the experimental setup, things like iron minerals or carbonates when using the CO2N2 atmosphere, actually neutralized harmful byproducts like nitrates, which tend to destroy amino acids.
So the rocks mattered.
Exactly. Adding these minerals significantly improved the amino acid yields again, suggesting that crucial interactions between the atmosphere, the ocean, and the planet's rocky surface were important. It wasn't just gases in the air. Even the borosilicate glass of Miller's original flask might have inadvertently acted as a silicate catalyst, mimicking the role of early Earth rocks.
That's a fascinating refinement. It wasn't just a soup. It was a soup interacting with the container, the early Earth itself.
It is, and here's where it gets really interesting. A real testament to preserving samples. Most astonishingly, in 2007, long after Stanley Miller's death, scientists, including some of his former students like Jeffrey Botta, reanalyzed Miller's original sealed vials from the 1950s.
His original samples, after 50 years.
Yes. Using modern analytical technology, techniques like high-performance liquid chromatography and mass spectrometry, which are literally 1 billion times more sensitive than what Miller had available back then.
1 billion times more sensitive. What hidden secrets did they uncover from his original experiments?
They discovered his experiments had actually produced a much wider variety of organic compounds than he could identify. They found over 20 different types of amino acids, including many used by life today that Miller hadn't reported. Far more than he knew.
Wow, 20. That's way more than he originally found. So his experiment was even more successful than he realized.
In many ways, yes. This confirmed the robust potential of these kinds of processes to generate life's building blocks, even if the exact starting atmospheric conditions were different from his initial guess. So while the specific recipe of the soup is still debated and refined, was it reducing, neutral, what minerals were key? The core principle that abiotic synthesis of complex organic molecules is possible under plausible early Earth conditions has been repeatedly validated and even strengthened over time. The debate has really shifted from if it could happen to where and how it happened most efficiently.
That's an incredible story of scientific progress, isn't it? That an experiment from the 50s still holds secrets waiting for better technology decades later. But even with this improved understanding of the soup's potential, there were still major conceptual challenges for this model to overcome, right? You mentioned things like the polymerization problem and the Homochorale puzzle. What are these profound hurdles?
These are indeed two profound challenges that highlight the limitations of just thinking about a dilute primordial soup in a vast ocean. The first is the polymerization problem. Even if you make lots of monomers, the building block's life needs long, complex polymers like proteins, chains of amino acids, and RNA, chains of nucleotides.
Right, you need to link them together.
Exactly. But the chemical reaction that links monomers together, called dehydration synthesis, as we discussed, releases a molecule of water.
Okay, and why is that a problem in an ocean environment? It seems intuitive that a wet environment would be ideal for chemistry.
That's the counterintuitive part. Because in a large body of water like an ocean, this reaction is thermodynamically unfavorable. Le Chatelier's principle tells us that if a product, water, is abundant, the reaction is pushed backward. The overwhelming tendency is for the reverse reaction, hydrolysis, the breakdown of polymers by the addition of water, to occur. The water actually attacks the bonds holding the chain together.
So the very medium that hosts the soup is chemically hostile to the crucial step of building the long chains needed for life.
Pretty much, yes. It's a major hurdle for the simple soup model. Proposed solutions generally move away from the open ocean and involve mechanisms for removing water.
Like those wet-dry cycles on land.
Exactly. Evaporation in shallow pools is a great way to drive polymerization. Or the catalytic and concentrating effects of mineral surfaces like clays, which can bind monomers and exclude water, facilitating chain formation.
Okay, so polymerization needs concentration and maybe drying. What's the other puzzle?
Homochorality.
The second major challenge is the puzzle of homochorality. Many essential organic molecules, including amino acids, except glycine and sugars, are chiral. This means they exist in two mirror image forms, like your left and right hand. They're chemically identical but structurally distinct, non-superimposable mirror images. We call these enantiomers.
Left-handed and right-handed versions.
Precisely. Now, abiotic chemical reactions, like those in the Miller-Urey experiment or likely occurring on the early Earth, invariably produce a 50-50 mixture of both left and right-handed forms. This is called a racemic mixture. But all life on Earth, It exclusively uses one specific handedness for each type of molecule, right?
Exactly. All life on Earth, without exception, exclusively uses left-handed amino acids,
Yeah.
L-amino acids, to build proteins, and right-handed sugars, D sugars, to build RNA and DNA. This is universal across all known life.
Why and how did that happen?
That's the mystery. How and why the specific handedness was selected from an initially random 50-50 mixture is a deep and fundamentally unsolved problem in origins of life research. The simple soup model offers no inherent explanation for this critical selection step. It's a fundamental asymmetry at the heart of biochemistry, and we don't fully understand its origin.
Okay, so the primordial soup idea, while groundbreaking, clearly wasn't the complete picture. It faced these big challenges like polymerization and homochirality. This led scientists to explore other possibilities, other potential cradles where life might have begun. And you mentioned the modern debate is largely framed by two competing or maybe complementary schools of thought: genetics first and metabolism first.
That's a good way to frame it, yeah. Though the lines are blurring now.
Let's start with the genetics first approach. This is often closely associated with the RNA world hypothesis, right?
Correct. The genetics first hypothesis posits that the first entity capable of undergoing Darwinian evolution, replication, variation, selection was an information-carrying molecule, a replicator. And the leading candidate for this role is RNA, ribonucleic acid, in a scenario known as the RNA world hypothesis.
And why RNA? What makes it so special compared to DNA, which carries our genes now, or proteins, which do most of the work in cells? Why could RNA have been the star of this early show?
RNA is uniquely versatile. It has this amazing dual capability. Like DNA, it can store genetic information in its sequence of nucleotides, providing the blueprint for life. But unlike DNA, RNA isn't just a passive blueprint. It can also fold up into complex three-dimensional shapes, much like proteins do, and act as a catalyst to speed up chemical reactions.
So it can be both the code and the machine.
Exactly. These catalytic RNAs are called ribozymes. This elegant solution addresses that classic chicken and egg problem that perplexed scientists for so long. Which came first, the genetic information, like DNA, or the catalytic function, like proteins? The RNA world hypothesis suggests RNA could have initially served both functions. Early life might have been based entirely on RNA carrying the genetic code and catalyzing its own replication and other essential reactions, with proteins and the more stable DNA evolving later to take over these specialized roles.
That really does seem to solve a big conceptual puzzle by giving RNA this dual identity. What kind of evidence actually supports this RNA world idea? Is it purely theoretical?
Oh no, there's substantial evidence that lends credibility to this hypothesis. A major breakthrough was the discovery of naturally occurring ribozymes in modern organisms back in the 1980s by scientists like Tom Seck and Sidney Altman, which earned them a Nobel Prize. This definitively proved that RNA catalysis is real and biologically relevant.
Like the ribosome. Isn't that involved in making proteins?
Precisely. The ribosome, the universal molecular machine that builds all proteins in all cells, is actually a giant ribozyme at its core. The catalytic activity comes from the RNA, not the protein components. That's a huge piece of evidence.
Wow, okay. Any other evidence?
Yes. Laboratory experiments, particularly those by researchers like Jack Szostak and Gerald Joyce, have successfully engineered ribozymes through directed evolution in the lab. They've created RNA molecules that can perform complex tasks, including accurately copying other RNA strands, sometimes even parts of themselves. This shows that RNA replication and evolution are chemically feasible.
So we can make RNA evolve in a test tube.
We can. Furthermore, many essential coenzymes and energy-carrying molecules in modern metabolism, like ATP, the cell's energy currency, and ADH, involved in electron transport, acetyl-CoA, they are all structurally based on RNA nucleotides, or contain parts that are RNA nucleotides. Many scientists view these as molecular fossils, remnants of a primordial RNA-based metabolism that preceded our current DNA protein world.
That's incredibly compelling, especially the idea of these molecular fossils still rattling around in our cells today. But what are the major challenges? The RNA world hypothesis sounds elegant, but it must face some formidable hurdles, right?
It absolutely does. Despite its elegance and the supporting evidence, the RNA world faces significant challenges. One of the biggest is the prebiotic synthesis of RNA's building blocks, the ribonucleotides themselves. Making these four complex molecules, A, U, G, C, and linking them to ribose sugar and phosphate groups under plausible early Earth conditions is chemically very difficult and remains a major, largely unsolved problem for chemists. It's much harder than making amino acids like Miller did.
So just getting the building blocks for RNA is tough.
Very tough. And even if you could make them, RNA itself is notoriously fragile and unstable, especially compared to DNA. It readily breaks down in water, hydrolysis, especially at higher temperatures thought common on early Earth, and it's easily damaged by UV radiation. So, making enough RNA and having it survive long enough to accumulate and start replicating is a serious problem. How do you protect it? How does it concentrate?
Okay, so the RNA world focuses on the information emerging first. But what about the metabolism first approach? Where do proponents of that idea think life truly began, and how did it work without genetics initially?
The metabolism first hypothesis takes a different starting point. It proposes that life began not with complex genetic molecules like RNA, but with simple, self-sustaining cycles of chemical reactions, a kind of primitive metabolism powered by a continuous flow of geochemical energy from the environment. In this view, genetic materials like RNA or DNA would have evolved later, perhaps as a way to stabilize, improve, and make heritable these successful metabolic networks. Energy and organization first, information second.
Interesting. And the most compelling location for this, you said earlier, is at specific types of deep-sea hydrothermal vents, the cooler alkaline white smokers. Why specifically these vents, and what makes their environment so uniquely suited for this idea?
Right, not the super hot black smokers, but the cooler ones, maybe 40, 90 degrees C, which are more conducive to complex organic chemistry. These are geological structures typically found near mid-ocean ridges where seawater circulates down through cracks in the Earth's crust, gets heated and chemically altered by interaction with rock, like olivine turning into serpentinite, and then is released back into the ocean.
Okay, so hot, mineral-rich water coming out.
Yes, but crucially, the vented fluid is alkaline, high pH, and rich in dissolved gases like hydrogen, H2, and methane, CH4, as well as sulfide ions. Meanwhile, the surrounding Haitian ocean water was likely mildly acidic, lower pH, due to dissolved CO2 and rich in dissolved iron. Now, the vent structures themselves are often made of porous minerals like iron, sulfides, and hydroxides, forming intricate chimney-like structures with countless tiny interconnected microcompartments or pores.
Like a mineral sponge.
Exactly, and these mineral walls physically separate the alkaline vent fluid inside the pores from the acidic ocean water outside. This setup creates a natural proton gradient, a difference in both pH, proton concentration, and electrical charge across the thin mineral walls of these microscopic pores.
Okay, hold on. That proton gradient sounds incredibly familiar. It's fundamentally how our own cells generate energy today, isn't it? Using something similar to a tiny electrochemical battery.
It is remarkably strikingly analogous to chemiosmosis, the fundamental energy harnessing mechanism used by every living cell today, from bacteria to humans. All known organisms pump protons, H+, across a membrane to create an electrochemical gradient, which they then use like water flowing through a turbine, the ATP synthase enzyme, to generate APP, the universal energy currency of life.
So the Venn hypothesis suggests life basically found a natural, pre-existing version of this energy system.
That's the core idea. Life simply co-opted this pre-existing geochemical energy source provided by the vent environment. This natural battery, the proton gradient across the mineral pores, could have driven the reduction of CO2 from the ocean water, using H2 from the vent fluid, forming simple organic molecules and kickstarting a primitive metabolism, all within the protected catalytic mineral pores of the vent. The energy source was built in.
That's a really powerful idea, having the energy source right there from the start. What are the key advantages of this deep-sea vent model compared to, say, the primordial soup, or even the surface-based RNA world?
It offers several key advantages. First, it provides a continuous, reliable source of both chemical energy, the proton gradient and reduced gases like H2, and raw materials, CO2 minerals, solving the energy problem inherent in the surface soup model, which relies on intermittent sources like lightning or UV.
Okay, continuous energy. What else?
Second, the deep ocean location offers complete protection from the harsh UV radiation and frequent, potentially sterilizing meteorite impacts that plagued the surface of the early Earth. This addresses the UV paradox directly. And finally, as we mentioned, the vent's honeycomb-like structure of mineral-walled pores provides natural compartments. This solves the dilution problem of the soup model by concentrating reactants and products, essentially acting as ready-made microscopic reaction vessels or inorganic cells.
That sounds incredibly robust.
Oh,
Continuous energy protection and natural concentration. It ticks a lot of boxes. But even the most compelling theories face scrutiny. What are the biggest conceptual or experimental hurdles the vent hypothesis still needs to overcome?
Well, while the energy mechanism chemiosmosis, driven by natural gradients, is highly plausible and supported by the universality of it in modern life, the spontaneous, non-biological formation of the specific, complex metabolic pathways and their key intermediates, like precursors to amino acids or nucleotides, solely within the vent environment has yet to be convincingly and completely demonstrated in laboratory experiments simulating those conditions. The chemistry is intricate, and getting it all to work abiotically is tough.
So we haven't fully replicated vent-based abiogenesis in the lab yet.
Not fully, no. There's progress, but it's complex. And perhaps the biggest conceptual hurdle remains explaining the monumental leap from a simple, geographically fixed, non-coded metabolic cycle confined to mineral pores to a free-living, replicating cell with a sophisticated, genetically coded system capable of evolution. How does a metabolism trapped in rock learn to encode itself, build its own membrane, and leave the vent? While the vent provides the potential energy and raw materials, the mechanism for inventing and encoding information is still a major puzzle for this model.
Okay, so we've got the RNA world focusing on information first, facing challenges with synthesis and stability, and the hydrothermal vent theory focusing on metabolism and energy first, facing challenges with demonstrating the complex chemistry and the leap to genetics. Are there other significant alternatives or perhaps complementary scenarios that scientists are exploring?
Absolutely. The field isn't strictly divided into just those two camps. One very compelling alternative, or perhaps complementary site, involves terrestrial hot springs. Think Yellowstone, but on the early Earth. These are dynamic, warm little ponds, a phrase Darwin actually used found on volcanic landmasses distinct from deep-sea vents.
So back on the surface, but still geothermally active.
Right. These geothermal fields provide rich chemical ingredients from volcanic activity and heat. But their key advantage, as we touched on earlier when discussing polymerization, is the prevalence of repeated wet-dry cycles. As pools evaporate, the concentration of monomers increases dramatically, and the removal of water strongly favors dehydration synthesis, the reaction needed to form polymers like RNA and peptides, short protein chains. Then, re-wetting, perhaps by rain or geyser splashes, allows these newly formed polymers to dissolve, move around, interact, maybe even self-assemble into more complex structures like protocells.
So that solves the polymerization problem very effectively.
It's considered a highly effective way to overcome the polymerization problem that plagues purely aquatic scenarios like the deep ocean or even a constant primordial soup lake. Some researchers, like David Deamer, argue strongly that these cycles were essential for the emergence of protocells with stable membranes encapsulating polymers.
Interesting. So instead of a deep-sea cradle, a more surface-level dynamic one, powered by geothermal heat in these crucial cycles. What about the idea of panspermia? That's always been a fascinating, almost sci-fi concept life, or its ingredients, coming from beyond Earth.
Panspermia is an ancient idea, actually, but it has a modern scientific basis, too. It suggests life isn't purely terrestrial in its ultimate origin. In its modern form, it proposes either that essential organic building blocks were delivered to early Earth from space, or, more radically, that fully formed microbes were transported here.
Delivery via what? Asteroids? Comets?
Exactly. Meteorites, comets, and even interstellar dust particles are known to carry organic molecules.
And is there concrete evidence for this delivery mechanism, at least for the raw materials, the building blocks?
Yes, there's very strong evidence for the delivery of raw materials. Numerous carbon-rich meteorites, like the famous Murchison meteorite that fell in Australia in 1969, have been analyzed and found to contain a rich variety of organic molecules crucial for life. We're talking amino acids, including some not found in terrestrial life, nucleobases, the letters of DNA and RNA, sugars, and membrane-forming fatty acids. These materials were definitely raining down on the early Earth.
So the ingredients could have come from space. What about whole microbes? Could they survive the journey?
That's more speculative, but experiments have shown that some incredibly hardy microorganisms, known as extremophiles, think bacteria like Deinococcus radiodurans or certain spores, can survive the vacuum, temperature extremes, and intense radiation of space for extended periods, at least when shielded within rock. So interplanetary transfer of viable microbes inside meteorites is at least conceivable, though proving it happened is another matter entirely.
Right. But even if life's ingredients, or even early microbes, came from space, does that truly solve the origin problem, or does it just, well, kick the can down the road, cosmically speaking?
That's its primary limitation as an explanation for abiogenesis itself. Panspermia doesn't actually solve the origin problem, how life first arose from non-life. It just relocates the problem to another cosmic location, Mars perhaps, or an exoplanet, or even interstellar space. It potentially explains how Earth got seeded with life or its components, but not how that life or those components originally arose anywhere in the first place. It shifts the question from how did life begin on Earth to the perhaps even harder question, how did life begin anywhere?
Okay, that makes sense. So considering all these different theories, RNA world, hydrothermal vents, terrestrial hot springs, the contribution from panspermia, are they strictly mutually exclusive? Or is there a converging, more integrated picture emerging in scientific thinking now?
That's a critical question, and I think the trend is definitely towards convergence. The genetics first and metabolism first views, and even the role of external delivery of materials, may not be mutually exclusive at all. Increasingly, researchers see them as potentially complementary aspects of a more complex process.
How so? How could they work together?
Well, for instance, a metabolism-first environment, like an alkaline hydrothermal vent or a geothermally active hot spring, could have provided the ideal cradle, the continuous energy, the protection, the mineral catalysts, the concentration mechanisms for a genetics-first system, most likely based on RNA, to be synthesized, stabilized, and begin to evolve. The geochemistry provides the reactor, and the RNA provides the information storage and replication needed for evolution to take off.
So a hybrid model. The vent or hot spring is the chemical factory, and the replicating RNA is its most successful product.
Exactly. In this hybrid view, the environment provides the energy and the protected chemical setting, overcoming RNA's fragility and synthesis problems, while the RNA provides the hereditary information needed to move beyond simple chemical cycles towards true Darwinian evolution. This kind of fusion seems increasingly plausible and helps resolve some of the primary paradoxes or weaknesses of each model when considered in isolation, like the energy source for RNA replication or the origin of information in metabolic cycles.
Okay, that integrated picture makes a lot of sense, combining the best features of different environments and chemical systems. So now you've got the building blocks, possibly delivered from space or made in situ, and some compelling potential cradles, vents, hot springs, where these chemical reactions could have become more complex, maybe even producing replicating RNA powered by local geochemistry. The next monumental step, then, is the actual emergence of the cell itself. How did chemistry truly turn into biology with the rise of the first enclosures, these things called protocells?
Right, this is a crucial transition. For life to advance beyond just being a random soup of interacting molecules, even replicating ones, those useful molecules, especially the genetic material and any catalysts helping it replicate, needed to be kept together. They needed to be prevented from just diffusing away into the vastness of the ocean or pond. The solution was compartmentalization.
Containing the chemistry.
Exactly, forming a boundary. This boundary structure was likely what we call a protocell, a simple precursor structure, not yet a full modern cell, but containing an informational polymer, like RNA, enclosed within some kind of rudimentary membrane.
And what formed these first membranes? Were they complex protein structures like in our cells today, or something much simpler that could form spontaneously?
Almost certainly something much simpler that could form spontaneously. The leading candidates are lipids, specifically simple fatty acids or similar amphiphilic molecules. As we discussed, these molecules have a water-loving hydrophilic head and a long, water-hating hydrophobic tail.
Right, the amphiphilic nature.
Yes. And in water, these molecules spontaneously self-assemble into structures that minimize the contact of their hydrophobic tails with the surrounding water. They can form spherical structures called micelles, like tiny balls with tails inward. Or, more critically for forming cells, they can arrange themselves into double layers, bilayers, which then close up into enclosed sacs called vesicles or liposomes.
Basically microscopic bubbles with a double-layered skin made of fatty acids.
That's a great way to picture it. These vesicles spontaneously trap whatever molecules happen to be nearby inside them when they form, so you get these tiny compartments encapsulating bits of the surrounding chemical environment.
Nature's own little test tubes, forming spontaneously just from simple fatty acids mixing with water. It's quite remarkable how fundamental chemistry provides such elegant solutions for life's beginnings.
It really is. You can imagine countless trillions upon trillions of these protocell vesicles forming constantly on the early Earth, especially perhaps in those environments with wet-dry cycles or near vents where lipids might be synthesized or delivered. Each one would create an isolated microenvironment.
A tiny experiment.
Exactly. A place where life's building blocks, maybe RNA strands and the nucleotides needed to copy them, could concentrate, leading to more frequent and efficient chemical reactions compared to the dilute ocean. And we even have evidence from space supporting this. The Murchison meteorite, for instance, contained fatty acids that, when extracted and simply placed in water in the lab, spontaneously formed stable vesicles, showing that these membrane-forming materials were definitely available on early Earth.
So the stuff to make membranes was there and self-assembles.
Mm-hmm.
And these early protocells weren't just static containers, were they? Did they have any primitive characteristics that we associate with living things like growth or division?
No, they were likely highly dynamic, not static bags. Their simple fatty acid membranes could actually grow by incorporating more lipids from their surroundings.
Yeah.
They could also be quite leaky, allowing small molecules like nucleotides, the building blocks of RNA, to diffuse in, providing the raw materials for an encapsulated genetic polymer to potentially replicate. And remarkably, these simple vesicles can even divide under purely physical forces, like shear stress from water flow, or if they grow too large, splitting into smaller daughter vesicles, distributing their contents in the process.
Wow. So primitive growth and division without complex biological machinery.
Yes, a very rudimentary form of reproduction. And this creates the potential for a crucial feedback loop, the beginning of Darwinian evolution at the protocell level. Imagine an RNA molecule trapped inside a vesicle that, purely by chance, had some catalytic ability to help synthesize more lipid molecules for the membrane. That protocell would be able to grow faster, incorporate more resources, and perhaps divide more readily than protocells without that helpful RNA. Its offspring vesicles would inherit copies of that beneficial RNA. This creates a link between the internal replicator, the genotype, and the success of the container, the phenotype, initiating a primitive chemical form of natural selection.
That's the key step, isn't it? Linking the information to the survival and reproduction of the whole package. Okay, so we have these very basic protocells, maybe with some replicating RNA inside, capable of primitive growth, division, and even selection. But then we often hear about something called LUCA, the last universal common ancestor. It sounds a lot more sophisticated than just a simple lipid bubble with some RNA. What exactly was LUCA, and where does it fit in the story?
LUCA is a crucial concept, absolutely. But it's really important to understand that LUCA is not the first life form. It wasn't the original protocell. Rather, the last universal common ancestor, LUCA, represents a much, much later and significantly more advanced stage of evolution. It's defined as the most recent ancestral population of organisms from which all life on Earth today, all bacteria, all archaea, and all eukarya, which includes us, ultimately descends.
So it's like the trunk of the tree of life,
Yes.
the single point back in time from which all existing branches emerged.
So that's a perfect analogy. Think of it as the ultimate great-great-great-grandparent population of every living thing on the planet today. It's the convergence point if you trace all current life backward.
Okay, so there's a common ancestor to all existing life, but presumably there was a lot of life, maybe even different kinds of life, before LUCA that went extinct or didn't lead to us.
Exactly. There was likely a long period of evolution, potentially involving many different types of protocells and early cells, before the lineage leading to LUCA became dominant.
How do scientists figure out what LUCA was like then? If there are obviously no fossils of this single-celled organism from billions of years ago.
That's a great question, and it's a real testament to the power of modern comparative genomics and bioinformatics. We infer LUCA's characteristics by looking for the genes and biochemical traits that are universally conserved, meaning they are shared by all modern life across all three domains, bacteria, archaea, eukarya. The logic is if everyone has it today, their last common ancestor must have had it too, having passed it down to all descendants.
So you reconstruct LUCA by finding the common denominator of all life today.
Precisely. And this genomic reconstruction paints a picture of an organism that was already surprisingly complex, far, far beyond a simple fatty acid protocell with just RNA.
What kind of complexity are we talking about for an organism that might have lived, maybe 3.8 or 4 billion years ago?
Well, based on these universal features, LUCA was certainly single-celled, and it likely already possessed a DNA genome, not RNA, as its primary genetic material. This implies the major transition from an RNA world to a DNA protein world had already occurred before LUCA, and this DNA was likely replicated by a DNA polymerase enzyme.
DNA already. And what about reading that DNA?
LUCA almost certainly used the universal genetic code, the same basic dictionary our cells use today, to translate genes encoded in DNA via messenger RNA, mRNA, into proteins. And it did this using a sophisticated machinery involving transfer RNA, tRNA, and ribosomes. Ribosomes themselves are incredibly complex molecular machines, and they're universal, suggesting LUCA had them.
Wow. DNA, ribosomes, the genetic code. That's a lot of fundamental machinery already in place. What about its structure and metabolism?
It definitely had a functional cell membrane based on lipids, probably a lipid bilayer similar in principle to modern cells, which maintained a distinct internal environment. Though some speculate its membrane might have been leakier or more dynamic than modern ones, perhaps still relying partly on external gradients like those found in vents. Metabolism-wise, Eleuthia was certainly an anaerobe, meaning it lived without oxygen, which makes perfect sense given the anoxic conditions of the early Earth we discussed. It was also likely a thermophile, thriving in hot conditions, perhaps 60–80 degrees C or even higher.
Living in the heat, no oxygen. What did it eat, or how did it get energy?
Eleuthia was probably an autotroph, meaning it could fix its own carbon from inorganic sources, likely from carbon dioxide dissolved in the water. The most likely metabolic pathway it used for this, based on universally conserved genes, is called the Wood-Jungdahl pathway, or acetyl-CoA pathway. This pathway is ancient, relies on hydrogen gas, H2 as an electron donor, CO2 as a carbon source, and importantly uses enzymes that contain iron-sulfur clusters as critical cofactors, hinting at a link to environments rich in those minerals.
Iron-sulfur clusters, like in those hydrothermal vents.
Exactly. And perhaps most significantly, Eleuthia generated its energy using chemiosmosis. It had protein complexes embedded in its membrane, including a rotor-stator-type ATPase enzyme, very similar to the ones in our mitochondria, which harnessed an ion gradient, likely a proton gradient, across its membrane to synthesize ATP.
So the very same fundamental energy mechanism we saw potentially existing naturally in the vents, Eleuthia was already using biologically.
Precisely. Those inferred traits of Eleuthia being a thermophilic anaerobe relying on H2, CO2, and iron-sulfur chemistry, and using chemiosmosis powered by proton gradients, they all point remarkably strongly towards an environment very similar to a deep-sea alkaline hydrothermal vent.
That convergence seems too strong to be a coincidence, doesn't it? Between what we reconstruct Eleuthia to be like and the conditions found at those vents.
It's anything but a coincidence to many researchers in the field. This striking convergence of evidence from comparative genomics, what Eleuthia was like in geochemistry, what vents are like, forms arguably the most powerful argument that the origin of the lineage leading to all life today was forged in, or at least passed through, these deep, dark, energy-rich environments. It's a very strong piece of circumstantial evidence for that kind of metabolism-first cradle supporting the early evolution of sophisticated cellular life.
Okay, but this means there was a huge evolutionary jump, an enormous gap between that simple protocell we started with, maybe just a lipid bubble with some replicating RNA and Eleuthia, this surprisingly complex organism with DNA, ribosomes, protein synthesis, and sophisticated metabolism.
Yeah.
You've called this the Great Leap. What does that immense, still poorly understood chapter in life's story actually entail? It sounds like the hardest part to figure out.
It is arguably one of the biggest black boxes in the story of life. The immense complexity of Eleuthia compared to a plausible starting protocell makes it absolutely clear that a profoundly revolutionary period of biological innovation and evolution occurred before Eleuthia existed. The journey from a simple protocell to Eleuthia's sophisticated DNA and protein-based machinery is one of the largest, most significant, and yet most poorly understood chapters in the entire history of life. It's truly a vast, uncharted territory that scientists are still actively working piece by piece to try and reconstruct.
What kinds of monumental innovations would have been absolutely required during this Great Leap? It sounds like a series of almost unbelievable inventions had to happen.
It would have required a whole series of incredible fundamental innovations, likely unfolding over hundreds of millions of years. Key among them would be, first, the RNA to DNA transition. This involved switching the primary genetic material from the relatively fragile catalytic RNA molecule to the much more chemically stable double-stranded DNA molecule for long-term information storage. This stability allowed for the evolution of much larger, more complex genomes.
Okay, swap RNA for DNA. What else?
Second, the invention of proteins as the main functional molecules. This required the evolution of the genetic code itself, the dictionary mapping nucleic acid sequences to amino acid sequences, and the incredibly complex ribosome machinery to actually translate that code. Once proteins took over most catalytic roles from ribozymes, it unlocked vastly greater biochemical diversity and efficiency, opening up huge new evolutionary possibilities.
Proteins are just much better catalysts for most jobs.
Generally, yes, much more versatile and specific. And third, a crucial step towards independence was the development of self-powered metabolism. Early protocells might have passively exploited natural energy gradients, like those in vents. But to become truly free-living organisms capable of colonizing diverse environments, cells had to evolve the ability to actively pump ions and generate and maintain their own electrochemical gradients across their membranes. This effectively allowed them to carry their power source with them, to escape the vent or the shallow pool and conquer the rest of the planet.
Those are three enormous steps: DNA, proteins, and self-generated power. It sounds like a lot to accomplish in sequence. Was it likely a single linear progression, one protocell lineage gradually getting more complex, or was it perhaps a messier, more communal process involving lots of different early life forms?
It's highly unlikely to have been a simple linear progression within a single lineage. A more plausible scenario, strongly supported by analyzing the patterns of gene distribution across modern genomes, is that early life existed for a long time as a global, interconnected community of diverse protocells or primitive cells. In this precellular or progenote stage, horizontal gene transfer, the swapping of genetic material between different organisms, rather than just inheriting it vertically from parent to offspring, was likely rampant.
So innovations could spread sideways through the whole community.
Exactly. An innovation like a better membrane lipid or a useful enzyme or even the transition to DNA could potentially spread rapidly through this network of exchanging genetic information. LUCA, in this view, may represent the point where a particularly successful combination of these core traits, DNA genome, ribosomes, core metabolism, chemiosmosis, etc., finally locked in and became vertically inherited within a stable cellular lineage. This event, sometimes called the Darwinian threshold, marked the point where vertical descent became the dominant mode of evolution and the familiar branching tree of life rooted in LUCA could truly begin.
That paints an incredible dynamic picture of early evolution, not just linear progress, but a whole community experimenting and sharing information. Now let's turn to how scientists actually find concrete evidence for this ancient, often microscopic world, long before complex organisms with shells or bones existed. How do we read the rocks themselves to see life's earliest fingerprints?
It's definitely challenging looking for signs of life billions of years ago, but there are several key lines of evidence. One of the most subtle yet powerful ways to detect the activity of ancient life is through analyzing carbon isotopes.
Carbon isotopes, how does that work?
Okay, so carbon naturally exists in two main stable forms, or isotopes. A lighter form, carbon-12, 12C, which makes up about 99% of carbon, and a slightly heavier form, carbon-13, 13C, making up about 1%. Now, biological processes, especially the enzymes involved in fixing carbon dioxide from the environment, like in photosynthesis or the Wood-Jungdahl pathway we mentioned for LUCA, tend to preferentially use the lighter 12C isotope. It's just slightly easier and faster for enzymes to grab and process.
So living things tend to incorporate more of the lighter carbon.
Exactly. This means that organic matter produced by living organisms becomes enriched in the lighter 12C relative to the heavier 13C, compared to the background inorganic carbon in the environment, like CO2 in the air or bicarbonate in the water. When these organisms die and their organic matter gets preserved in rocks, often as graphite or kerogen, this isotopically light signature is preserved too.
So if you find ancient carbon in a rock that's significantly enriched in this lighter carbon-12, it's a strong chemical sign, a biosignature of past life.
Precisely. A significantly isotopically light carbon signature in ancient rocks is considered a strong potential biosignature. Geochemists carefully measure the ratio of 13C to 12C in these samples. The oldest rocks where this kind of isotopically light carbon signature has been found suggesting biological activity are in the Isua Greenstone Belt in southwestern Greenland. These rocks are dated to approximately 3.85 billion years ago.
3.85 billion years. That's incredibly early, really pushing back the evidence for life almost as far back as the oldest known rocks themselves. It suggests life emerged remarkably quickly once conditions became even marginally hospitable after the Hadean bombardment perhaps settled down.
It really does suggest a rapid emergence. Now, it's important to be cautious. It's not definitive proof on its own, as some non-biological processes under very specific conditions might theoretically mimic this isotopic fractionation to some extent. But it's the earliest and one of the strongest chemical hints we have that life's metabolism was active very early on, potentially within just a few 100 million years of the oceans forming.
Okay, so isotopic signatures give us chemical hints. But what about actual physical fossils? When do we start seeing structures that look like they were built by life?
For direct physical evidence, we look to structures called stromatolites. These are fascinating.
Ah, the living rocks. Many people might recognize that name or have seen pictures of them. What exactly are stromatolites, and how do they give us a window into early life?
The stromatolites are not actually fossils of individual microorganisms. Instead, they are layered, often mound-like or dome-like, sedimentary structures that were built by entire communities of microorganisms, primarily photosynthetic microbes like cyanobacteria in later geological times, or possibly earlier anoxygenic photosynthesizers.
So, microbial communities building rocky structures layer by layer. How do they do that?
Imagine a shallow water environment. These microbes form sticky mats or biofilms on the sediment surface. As sediment particles suspended in the water settle down, they get trapped by this sticky microbial layer. To avoid being buried, and crucially for photosynthetic microbes to stay in the sunlit zone, the microbes then migrate upwards through the trapped sediment, forming a new layer on top. They also often precipitate minerals like calcium carbonate, which helps cement the layers together.
And over time, this builds up.
Over millennia, this continuous process of trapping sediment, growing upwards, and cementing the layers builds up these distinctive, finely laminated rock structures. They can range from small CM-scale domes to massive reef-like structures meters high.
And you can still see these forming today, can't you? In a few very specific, often harsh environments on Earth.
You absolutely can. In a few places, with conditions that discourage grazing animals which would normally eat the microbial mats, typically hypersaline lagoons like Shark Bay in Western Australia, or some alkaline lakes, you can find modern, living stromatolites being built by microbial communities today. And they look remarkably similar in their structure to their ancient fossil counterparts, providing a direct analog and confirming the biological process involved in their formation.
That's amazing. So how old are the oldest fossil stromatolites that scientists generally agree on?
The oldest widely accepted stromatolite fossils showing clear biological layering and morphology were found in the Pilbara region of Western Australia. These are dated to approximately 3.5 billion years ago.
3.5 billion. So by that time, life wasn't just present as maybe individual cells leaving chemical traces, but it was already complex enough to form organized, cooperative ecosystems, microbial mats, that were actively interacting with their environment and building large-scale geological structures.
Precisely. It shows a significant level of biological organization and environmental interaction had already evolved by 3.5 Dre. That roughly 350 million year gap between the earliest chemical isotopic signatures in Greenland, around 3.85 Dre, and these first widely accepted physical fossils in Australia likely represents a crucial period of invisible evolution, where life perhaps existed mainly as free-floating single cells before developing the ability to form these large mat-building communities.
Okay. And speaking of life actively shaping its environment, that brings us to arguably the most profound example of this in Earth's history, the Great Oxidation Event, or GOE.
Ah, yes, the GOE. This is where life didn't just adapt to the planet, it fundamentally changed the entire planet on a global scale.
It terraformed Earth, essentially, from the oxygen-free world where life began to the oxygen-rich atmosphere we depend on today. How did that monumental shift happen? What triggered it?
For well over 1 billion years, maybe closer to 1.5 billion years after life first arose, Earth remained essentially an anoxic world. Early forms of photosynthesis used by many bacteria didn't produce oxygen as a byproduct. They used things like hydrogen sulfide or iron instead of water as their electron source. But then a truly revolutionary metabolic innovation evolved, likely within a group of microbes we now call cyanobacteria.
Cyanobacteria, sometimes called blue-green algae.
Right, they invented oxygenic photosynthesis. This new type of photosynthesis used the incredibly abundant molecule water, H2O, as the source of electrons, splitting it apart, and the byproduct of splitting water is free oxygen, O2.
So cyanobacteria started pumping out oxygen gas as a waste product into the environment, a pollutant from their perspective.
Exactly, a highly reactive, potentially toxic waste product. But it didn't just immediately build up in the atmosphere, did it? What happened to all that early oxygen being produced, perhaps for hundreds of millions of years before the GOE? That's a key point. For a very long time, perhaps starting as early as 3 billion years ago or even before, this biologically produced oxygen didn't accumulate significantly in the atmosphere or surface oceans. It was immediately consumed by reacting with vast quantities of dissolved ferrous iron, Fu2+, that were abundant in the anoxic oceans at the time. Oxygen oxidizes ferrous iron into insoluble ferric iron, Fe3+, which then precipitates out of the water as iron oxides, essentially, rust.
So the oceans were literally rusting on a massive scale.
On a planetary scale, yes. This massive precipitation of iron oxides settled down onto the seafloor. This process is recorded globally in the geological record as the world's most impressive and economically important iron ore deposits, the Banded Iron Formations, BIFs.
The BIFs. Those distinctive rocks with alternating bands of iron oxides like hematite or magnetite and silica-rich layers like chert or jasper.
Precisely. These thick, extensive BFs were deposited primarily between about 3.0 and 1.8 billion years ago, and they represent a gigantic geological signature of cyanobacteria producing oxygen, and that oxygen then reacting with dissolved iron in the oceans. They are essentially fossilized rust, showing the planet's oxygen sinks in action.
So the oceans acted as a huge buffer, soaking up the oxygen produced by cyanobacteria for maybe hundreds of millions of years. What happened when those oceanic iron sinks finally got, well, full, when most of the dissolved iron had rusted out?
That seems to be the tipping point. Around 2.4 billion years ago, give or take 100 million years, these oceanic iron sinks became largely saturated. There just wasn't enough dissolved iron left to react with all the oxygen being continuously pumped out by the cyanobacteria. With nowhere else to go, free oxygen finally began to escape the oceans and accumulate in the atmosphere for the first time in Earth's history. This triggered the Great Oxidation Event, GOE.
The moment Earth's atmosphere started to contain significant free oxygen, that must have been absolutely catastrophic for the vast majority of life that existed at the time, which was anaerobic, adapted to an oxygen-free world.
It was transformative, and for many organisms, yes, catastrophic. Oxygen is a highly reactive molecule, toxic to organisms not adapted to handle it. It causes oxidative stress, damaging DNA and proteins. The GOE likely led to the first great mass extinction event in Earth's history, wiping out vast swaths of the previously dominant anaerobic microbes.
A poison gas crisis caused by life itself.
In a way, yes. Rising oxygen also had other major environmental consequences. It reacted with methane, CH4, a potent greenhouse gas that was likely more abundant in the early atmosphere, removing it. This reduction in greenhouse warming may have plunged Earth into a series of global glaciations known as the snowball Earth events around that time.
Wow, so mass extinction and possibly global ice ages, all triggered by microscopic cyanobacteria changing the air.
It sounds dramatic, and it was, yet this crisis also opened up incredible new evolutionary opportunities. The presence of atmospheric oxygen eventually led to the formation of the protective ozone layer, O3, in the upper atmosphere, which shields the Earth's surface from harmful UV radiation. This shielding was likely crucial for life to eventually colonize land surfaces successfully.
And maybe most importantly.
And perhaps most importantly, the availability of oxygen allowed for the evolution of a new, far more efficient way to generate energy from organic molecules: aerobic respiration. Using oxygen as the final electron acceptor in metabolism releases vastly more energy than anaerobic pathways. This metabolic revolution powered the rise of larger, more complex cells, the eukaryotes, and eventually multicellular life, including animals like us. The GOE, driven entirely by biology, is the ultimate demonstration of life's incredible power to not just adapt to its planet, but to fundamentally re-engineer it on a global scale, setting the stage for the entire subsequent evolution of complex life.
What an absolutely incredible journey we've taken today. Starting from a fiery, alien Hadean Earth devoid of life, through the first tentative chemical steps, the emergence of replicating molecules in protocells, eventually forming that sophisticated single cell, LUCA, which gave rise to every living thing you see today, and then even re-engineering the planet.
It truly is a mind-boggling story, pieced together from so many different fields: geology, chemistry, biology, physics. And the scientific consensus has definitely moved far beyond just a simple primordial soup in a static ocean. A much more nuanced, dynamic, and integrated picture is emerging now, one that acknowledges the strengths and potential contributions of multiple hypotheses and environments, weaving them together.
So let's try to synthesize this converging picture as we understand it now. It seems to begin with a young Earth, its atmosphere and oceans being seeded with simple organic molecules, the monomers. These are likely synthesized both terrestrially, driven by energy sources like lightning and UV radiation,
and importantly, also delivered from space via meteorites and comets, providing a potentially significant external source of building blocks.
Right. Then, crucially, the next step, polymerization, building the complex chains like RNA and proteins, probably occurred not just anywhere in a dilute global ocean, but specifically where these monomers could be highly concentrated and assembled.
Exactly. The leading candidates for these energy-rich, concentrating microenvironments remain those porous mineral structures of deep-sea alkaline hydrothermal vents with their built-in chemical gradients and those dynamic clay-lined geothermal pools on volcanic landmasses driven by their powerful wet-dry cycles. Both offer plausible solutions to the concentration and polymerization problems.
And within these geochemical reactors, perhaps a genetics-first system, most likely based initially on the versatile RNA molecule, could have emerged and started replicating. But its synthesis and replication might have been critically dependent on and powered by the continuous energy flow and chemical environment of its metabolism-first surroundings. That hybrid model really seems to resolve some of those primary paradoxes we discussed, doesn't it?
That's the real power of the integrated view, I think. It suggests the environment provided the necessary energy and protection, overcoming RNA's inherent fragility and synthesis challenges, while the RNA provided the crucial information storage and replication capability needed for Darwinian evolution to actually begin. It's a synergy between geochemistry and biochemistry.
Okay, so these first replicating systems, likely RNA-based, then become encapsulated within self-assembling lipid membranes, forming the first protocells.
Right. And then through potentially hundreds of millions of years of countless trials and errors involving intense competition and cooperation, likely fueled by rampant horizontal gene transfer within a diverse community of these primitive entities, life evolved, adapted, and innovated. This eventually gave rise to the much more sophisticated DNA and protein-based machinery we infer for the last universal common ancestor, LUCA.
And once LUCA's descendants, the ancestors of bacteria and archaea, escaped their potential cradle, perhaps the vents or hot springs, they began to diversify and spread, fundamentally re-engineering the planet's biogeochemical cycles, culminating eventually in the Great Oxidation Event.
Which in turn set the stage for the evolution of eukaryotes and the complex macroscopic world we know today. It's truly a story of planetary scale engineering, driven initially by microbes, shaping the very air we breathe and the ground beneath our feet.
It really is. But, as you've stressed, despite all this incredible progress in understanding the outlines of the story, humility is essential. The origin of life remains one of science's most formidable challenges, doesn't it? It's driving vast amounts of current research precisely because there are still huge unanswered questions.
Absolutely. We've sketched a plausible narrative, but many of the crucial details are still fuzzy, debated, or simply unknown. There are fundamental mysteries that keep scientists in this field awake at night.
What are some of those biggest remaining questions, the frontiers that scientists are still grappling with at the cutting edge of origins of life research?
Well, perhaps the deepest mystery is what's often called the information problem. How did the specific, meaningful sequences of nucleotides, the genetic information that actually code for functional molecules like ribozymes or later proteins, arise from essentially random chemical processes? How does sequence generate function spontaneously? And related to that, the origin of the genetic code itself. The seemingly arbitrary but universal dictionary that translates nucleic acid language into protein language is almost entirely unknown. It's like finding a complex instruction manual written in a language with no Rosetta Stone.
How did the code come to be? Yeah, that's profound. And the homochirality problem you mentioned earlier, why life uses only left-handed amino acids and only right-handed sugars, that's still a major puzzle, isn't it, given that prebiotic chemistry likely produced a 50-50 mix?
It remains a fundamental unsolved mystery. There are hypotheses involving polarized light or mineral surfaces or amplification mechanisms, but no definitive consensus on how this universal biological preference was established from an initially racemic prebiotic world. And then there's the huge gap we discussed, the protocell to LUCA transition, the detailed evolutionary pathway, the specific intermediate steps, from a simple vesicle containing maybe some replicating RNA to the astonishing complexity of LUCA, a DNA-based cell with ribosomes, protein synthesis, sophisticated metabolism, the works that's still largely a vast uncharted territory. Reconstructing these crucial steps is a primary goal for many researchers.
It feels like reconstructing the evolution of the engine while only having the blueprint for a wheel in the final car.
That's not a bad analogy. And finally, there's a more philosophical but also practical challenge, the inherent grayness of life. Abiogenesis wasn't an instantaneous event. It was a gradual process, a continuum from complex, non-living geochemistry to simple living biochemistry. This creates a fundamental ambiguity. Where exactly along that continuum do we draw the line and declare something definitively alive? This fuzziness complicates not only our definitions, but also our strategies for detecting very early life signatures on Earth or potentially simple or nascent life elsewhere in the universe.
That's a really interesting point. So given these profound remaining questions, where is the research heading now? What are the most exciting future directions and origins of life research? How are scientists trying to crack these tough nuts?
The path forward is as interdisciplinary as the field's history, combining lab experiments, theoretical modeling, geology, and even astronomy. One major direction is increasingly looking outward, leveraging the field of astrobiology.
Looking for life, or its precursors, beyond Earth.
Exactly. The exploration of potentially habitable environments in our own solar system, like Mars, which had water in its past, or the subsurface oceans of icy moons like Jupiter's Europa and Saturn's Enceladus, offers the tantalizing possibility of discovering extraterrestrial life. Or perhaps more likely, initially, discovering prebiotic chemistry in action or relics of it. The potential discovery of active hydrothermal vent systems on Europa or Enceladus, for instance, could provide a direct natural laboratory to test the vent hypothesis for life's origins.
It's incredible to think that studying chemistry on a moon orbiting Saturn could directly inform us about how life started here on Earth billions of years ago.
It's a powerful connection, and the fact that life appears to have emerged on Earth relatively quickly, geologically speaking, once conditions became somewhat habitable, suggests to some statisticians that abiogenesis might not be an astronomically improbable fluke. Maybe it's a relatively common process on Earth-like planets throughout the cosmos. This lends a certain optimism and urgency to the search for life elsewhere.
Okay, so looking outward with astrobiology. What about looking inward here on Earth?
Simultaneously, the search turns inward, pushing the boundaries of synthetic biology. Scientists are no longer just simulating presumed prebiotic conditions in flasks like Miller did. Now they are actively attempting to construct novel chemical systems from the bottom up in the lab, trying to build artificial life, or at least lifelike systems.
So literally trying to build simple self-replicating cells from scratch.
That's the ultimate ambitious goal for some researchers, yes. By trying to design and build simple, self-replicating vesicles containing evolving RNA molecules or other informational polymers in the lab, researchers can directly test hypotheses about how the crucial gaps, like the gap between non-living chemistry and self-sustaining evolution, or the protocell to LUCA transition, might have been bridged. The aim is to demonstrate experimentally a plausible step-by-step pathway from simple non-living molecules all the way to a self-sustaining, evolving protocell. Achieving that would be a monumental step towards finally providing a comprehensive, experimentally grounded answer to one of humanity's oldest and most profound questions. Where did we come from?
What an incredible scientific quest. We've covered so much ground today, tracing this amazing story from a fiery alien Hadean Earth through the complex chemistry that led to the first replicating systems, the emergence of cells, LUCA, and ultimately every living thing around us, including ourselves. The quest to fully understand the origin of life is truly one of humanity's greatest scientific endeavors, constantly evolving with new discoveries, new technologies, and new ideas. It's a powerful testament to human curiosity and ingenuity.
It really highlights how interconnected everything is, from the stars to the rocks to the very cells within us.
Absolutely. So as we wrap up, here's something for you, our listeners, to ponder. What does this deep dive tell you about our place in the universe? Knowing how quickly life might have emerged here on Earth, perhaps just as soon as conditions stabilized enough. What does that suggest to you about the likelihood of life arising elsewhere in the vastness of the cosmos? Is the universe teeming with potential life, or are we truly alone? Something to think about until our next deep dive.